I am Mageshwar. We saw about somaclonal variation. Somaclonal variation. It is a genetic variation in plants that have been produced by plant tissue culture and can be detected as genetic or phenotypic traits. Basic features of somaclonal variation. Variation in number and structure of chromosomes are commonly observed. Regenerated plants with alternated chromosomal changes often show changes in leaf shape and color, growth rate and habit, and sexual fertility. It is generally heritable mutation and persists in plant population even after plantation into the field. Mechanism of somoclonal variation. Genetic heritable variation. Pre-existing variation in somatic cells of X plant. They caused by a mutation and other DNA changes occur at high frequency. Epigenetic one. It is non-heritable variation. Variations generated during tissue culture caused by temporary phenotypic changes occur at low frequency. Callus tissue organogenesis regenerated plants, hardening and self-ning of somoclonal variant steps involved in induction and selection of somoclonal variations. Physiological causes. Exposure of culture to plant growth regulators, culture conditions. Genetic causes. Changes in chromosome number. Aneuploidy, gain or loss of one or more chromosome. Polyploidy, gain or loss of entire genome. Translocation, arms of chromosome switched, inversion, piece of chromosome inverted. Change in chromosome structures are deletion, inversion, duplication, translocation. Gene mutations are transition, transversion, insertion, and deletion. Plasma gene mutation, transferable element activation. DNA sequence. DNA change in reduction of alternated fragment size by using restriction enzyme. Change in protein, loss or gain in protein band, alternation in level of specific protein. Methylation of DNA. Methylation inactivates transcription process. Biochemical causes. Lack of photosynthesis, ability due to translation in carbon metabolism. Biosynthesis of starch via carotenoids pathway nitrogen metabolism. Antibiotic resistance. Advantages of somoclonal variations. It helps in crop improvement, creation of additional genetic variations, increased and improved production of secondary metabolites, selection of plants, resistance to various toxin herbicides, high salt concentration and minerals toxicity, suitable of breeding of tree species. Applications to crop improvement, improvement of existing clonal structures in sugarcane, selection of higher yielding and decrease disease resistance. Potatoes, yield and disease resistance, improved generium species, centered varieties, woody ornamental, Paulvinia selection of leaf variations. Disadvantages of somoclonal variation. A serious disadvantage occurs in operation which requires clonal uniformity in horticulture, and tissue culture is employed for rapid propagation of allied genotypes. Sometimes it leads to undesirable results. Selected variants are randomly and genetically unstable. It requires extensive and extended field trials, not suitable for complex ergonomic traits like yield quality, may develop variant with phenotypic effect which are not true. Thank you.